Breathe it out. Okay. Sorry, we're back. All right. This way.
So, like Ola said in previous lectures last week, we read the evidence from the evidence receiving. Evidence beginning with an amino acid change, the amino group of the first amino acid change, which is this nitrogen and the 3H organ with plus ones, and the C for this basically at the end, Ca2O. And so when you read an amino acid, read from left to right.
And then what bond is this?
Between C and
Between the amino acids. Yep. Yeah, so your peptide bond is gonna connect all your amino acids. And then when you're reading it, like Austin said, from left to right. Okay, so some questions to keep you awake. What is a protein made of? That was it? That was, why do you say ribosomes?
Because. Oh, wait, no, ribosomes are made of protein.
Yeah, that was perfect.
Oh,
Ribosomes are made of,
yes.
ribosomes are made of protein, and then the proteins are made of these like strings of amino acids.
Oh,
So that's what a protein is, and then proteins are synthesized by the ribosome, so they're translated by. And then what catalyzes the formation of these bonds? Like, where does this happen? What did you say? Yeah, the ribosome. Sorry, I did it away. Sorry. And then what kind of bond is a peptide bond? Like, what does it fall under? Yeah. And then through what reaction is the peptide bond formed? And what is the byproduct of this reaction? Mhm. Exactly, yeah, that kind of contributes to the aqueous environment of the cell. And then in a protein of 120 amino acids, which terminus is the 100th amino acid closer to?
on the end, so the C terminus.
Yep, perfect, nice. And then the answers for your personal review.
Hey, chemistry. This is fun. And Valentine's, I really like this one. It makes a lot of sense once you understand it. And so, first thing first, I wanted to touch upon the idea of electronegativity. So electronegativity basically means how strong an atom can attract electrons. And so the stronger the electronegativity, meaning the higher the electronegativity number that you pass on the electronegativity table, which is this one. So these are the very electronegative elements, and yeah, and they are very good at attracting electrons, and the three that you guys will see a lot is carbon, nitrogen, and oxygen. So high electronegativity, a high difference in electronegativity will play a role in the bond that I will talk about. So, covalent bond. So the key idea about covalent bond I want to point out is that one is that it's a type of intramolecular forces. Intramolecular forces meaning forces and bond that happens within a molecule. And the other thing about covalent bond is that it's electron sharing between two atoms. And so, that's what I said, like non-covalent bond is charge interactions, and covalent bonds are electron sharing. So I really want to point that out. So there's two types of covalent bond. The first one is polar covalent bond. Like I said earlier, big difference in electronegativity results in a polar results. For example, oxygen and hydrogen. So there is a lot of sources on the internet that shows like the electronegativity number of elements. I just put it in here so we have it. Oxygen has 3.5, which is very high. Hydrogen has 2.1 and the other one 2.1. And so the difference in the electronegativity is 1.4, which is a lot. And so the key thing here is that since the difference is very high, it means that oxygen attracts electrons more than its side, on its side than hydrogen is. I will explain that. Have you guys, is any one of you taking chem? 111, 112? Okay, so I just want to cover this. Oxygen, basically all types of, a lot of elements wanted to achieve a thing called the octet rule, meaning that it wants to have at least 8, max 8 valence electrons on the in its outer shell, and so oxygen has 6. 1, 2, 3, 4, 5. 2, 3, 4, 5, 6. So it wants 2 more, right? And hydrogen, it's like a very special case. It wants only 2, but each has 1. And so what they do is that these 2 share electrons together so they could achieve what they want. So oxygen will get 8 and hydrogen will get 2 for each. And so 1 for them, and then what they will do is that the hydrogen will come here and it will share 1 electrons, and the other hydrogen will come here and share another electrons. And what this will do is that these 2 electrons will be shared together with oxygen, and so oxygen will end up having 8, and each hydrogen will end up having 2. And so this is like what they really wanted. And this sharing of electrons, it's covalent bond. And so now we got that. Does anybody have a question?
So hydrogens share electrons with each other?
Yeah, hydrogens share electrons. Each of them contribute to this whole bonding thing. So each hydrogen has 1. They bring their electrons together with the O. O has 6. And so it's like a place for them to share together. And so those 2 electrons bring here and they ended up having 2 for each. Like the electrons move around. And so they ended up having 2 on the valence shell. And the oxygen ended up having 8, which satisfied both sides. And so this sharing is covalent bond. Okay. So like I said, oxygen has a very high electronegativity and hydrogen has a very low electronegativity. What that means is that these electrons will be, they run around. Like I draw this, but like, they actually run around, like right now, but they do run around, but like since electron has more electronegativity, it means that the electrons will be more likely on this side. It will stay on this side more. And since electron is negatively charged, this will create a partial negative charge on the oxygen side and a positive partial negative charge on hydrogen side. And this creates a polar bond. And so these 2 partial charges creates a polar bond. Anybody have a question? So with that same idea for non-polar covalent bond, there's little there's little to no electronegativity difference. And so I have here chloride, each chloride ions of the atom has 3.0 and they are the same. And so basically there's no difference in there. And so they share electrons equally. And so there's no partial charges on either side of the molecule. And this is called, that's why it's called non-polar, because there's no poles on either side. And they still share electrons, but no pole. No. So. We have covered, covalent bonds, now we're gonna do non-covalent bonds. Non-covalent bonds are charge interactions. There's no electrons involved. There are charge interactions and they happen between molecules, not within a molecule. And so first with ionic bond is formed between two ions. A full positively charged ion and a full negatively charged ions. An example for this is NH3 plus. So normal, this ion has different forms. It has NH2, NH, 4, NH4 plus. This form has a positively charged ion. It means that it's like positively charged by nature. So it doesn't form some sort of bond or anything. It loses, it gains electrons, so that's why it loses electrons, so that's why it has the charge. An example for negatively charged ion is fluoride ion. Basically, it means that it loses one electron by nature. That's why it has this negatively charged. An ionic bond is a bond that forms between the two. It's a charge interaction, meaning it's positively charged and negatively charged, and so they attract each other. So it's opposite attracts. And so for dipole interactions happen between two polar molecules. So for example, it will be two water molecule. Yeah, and so like I said, the oxygen side is more partially negative than the hydrogen side. And so what this does is that the partially negative side of this water molecule interact with the positively charged side of the other water molecule, which is the hydrogen part of it. And that is called a dipole-dipole interaction. And this is a hydrogen bond. Basically, what hydrogen bond means is that the bond between hydrogens, usually between hydrogen, oxygen, hydrogen, fluorine, or something else, but one example is this. And the last one is which can be very confusing. But before I touch upon that, you guys have any questions regarding this?
So are all covalent bonds intramolecular forces and all non-covalent bonds are intermolecular forces?
Yeah.
Okay. Even like ionic bonds?
Ionic bond is like ionic bond is intra because it's like it's kind of in the weird place because it happens between two ions. It's and each ion can be a molecule, but it can be a single atom. If you
So all covalent bonds are intra and then ionic bonds are also intramolecular? But then dipole van der Waals are intermolecular.
yeah, intermolecular.
Okay.
So these two would exist between molecules. This is usually happening within a single molecule, and covalent bonds are happening within a single molecule.
You can also have intermolecular ionic bonds, like this will happen, I'll talk about it later in tertiary structure, but like one amino acid that has a positive charge is interacting with another amino acid that has a negative charge. Those are two separate molecules, but they're ionically bonding. Does that make sense?
Because like the thing about ionic bond is that it can form between an ion that is a molecule. And so typically it's intermolecular forces for that case. But if ionic bonds form between, for example, Na plus and Cl minus, which are two atoms, then it's intramolecular forces. So the last one is kind of tricky. And so I'll try to explain. So first diagonal walls are basically interactions that happen between two non-polar molecules. It means that two non-polar molecules I've mentioned this, for example, ClCl Oh my god. CL. The first thing I want to mention is that these ions have like, these things, these atoms have electron clouds around them. And so they have electrons running around. That's the first thing I want to mention. Okay, now, like if it's stood, for example, if it's stood here by itself, here it's the same, like nothing happens. But if there's this molecule like run across this, the interaction happens, which basically means that the electrons that are running around on this fluoride molecule started to repel the electrons running on the other molecule. And this repel caused something called repolarization, meaning that the electrons start to go over on the other side. And so it can run here, but then they start to repel each other, and so it starts running over here. And what this does is that it creates a partial negative on this side. And the side that the electron ran away is partially positive. It happens the same for both molecules, so it induced a dipole molecule from a nonpolar molecule. Because the electrons repel each other as they move closer to each other. Does anybody? Yeah.
So why do they repel exactly? Is it
Um,
because-
So atoms have electron clouds. What that means is that electrons can be anywhere on this cloud. And so, for example, if this electron is on this side over here, and as there's another like atom over here that runs across by, and it has its electron like running closer to here as well, it repels each other because it's like the same charges. Yeah, so they came here together and they were like, no, I hate you, and then they started going on this side. Yeah, and that creates a partially positive on the side that has no electrons because it started running over here, and a partially negative on the side that the electrons started running. Cool.
They were going to show you, why is the body wall interactions?
I'm sorry.
Because referring forward, the non-phosphorescent, the non-forward, the middle ones are in the middle. So that's a part of the structure. So they repel each other. They stick to it.
I haven't touched that part yet. Okay, so now, so the thing, yeah, you got that. And so the next thing is that these polar side, this side of the partial negative charge of this molecule start to attract this side, the partially positive charge. This reaction repel electrons result in a dipole. That dipole start to interact with each other because it's opposite charges. Yeah, so like in a cellular environment, there's molecules everywhere. And so one molecule make this molecule dipole and another molecule make another dipole. And so maybe there's like another molecule here that has the partially positive charge over here. They're charged together. You get it.
Where do the London dispersion forces fit?
London dispersion forces and variables are falling under the same category. And so they can be understood as.
So they're like kind of one and the same?
Mm.
Okay.
Do you have a question?
Oh, yeah, so electrons.
Oh, so,
Yeah, this is like an example of entry.
Okay.
For metal.
I'm gonna take over. Okay. All right, So finishing up cell chemistry and bond, this is just explicitly stating what hydrophobic and hydrophilic is. Your hydrophobic molecules, like we mentioned, they're kind of gonna agglomerate together, they're gonna stick together, they just don't want to interact with polar environments, kind of hydrophobic and water fearing. And then hydrophilic, water loving, and polar interact with the aqueous polar environment. This is a phospholipid bilayer, has 2 layers, hence the name bilayer. Your polar heads, the X one comes up, I don't know. The red X. Your head, so that's extracellular, so outside of the cell, super aqueous, watery. You're gonna need that to be polar to interact with all these molecules. And then your hydrophobic part is gonna be your fatty acid chain. And then they're gonna face the hydrophobic part of the. And then inside the cell, super aqueous, super watery, you need that to be polar as well. And then when you have something like a transmembrane protein, you're gonna want this part to also be hydrophobic, that middle part. And then your heads are usually polar to interact with a substance that needs to signal. This usually happens with like cell signaling, so like something will bind to the receptor and lead to signaling in the cell, so that head needs to be polar. And then there's a sliding scale from hydrophobic, hydrophilic. Acids usually aren't 100% hydrophobic or 100% hydrophilic. They have characteristics about them. They have certain bonds that they have that are hydrophobic. Usually your carbon hydrogen bonds are going to be super non-polar, super hydrophobic. If you've ever seen like your amino acids that like hexagon shape, that's with a circle in the middle, that's a benzene ring. Which is just a lot of carbon and hydrogen, gonna be hydrophobic. And then you have your atoms like oxygen, even sulfur, we consider to be a little bit, hydrophilic and polar, nitrogen, stuff like that. So you just have to look at your amino acid in question, look at what properties it has, look at what atoms it has, and then classify if something is gonna be more hydrophilic or more hydrophobic based on the bonds that it has. Okay, so this is a little bit about protein modeling. I'm just gonna go through them quickly. Ball and stick and wireframe. That's what it looks like. Your ball is gonna be the atom, and your sticks are the bonds between the atoms. It shows every single atom and covalent bond in the protein. So it has your R groups and your backbone. It's just hard to discern what is what. So that's your con for that model. And then wireframe is the same thing as ball and stick, but it just shows those bonds without the atoms. And then you have your spatial model that also shows the atoms and then their bonds, and it physically takes up space in the image. This is nice so you can kind of see like the size of your protein, things like stuff related to the size and how big it is, how much space it takes up, but you can't really see the secondary structure or the backbone of the protein. You can usually see those R groups pretty well, but it's really hard to see the backbone. And then ribbon model is almost exclusively used for seeing secondary structure. So there's a lot of alpha helices in that picture. Those little swirls are your alpha helices, and then you'll have a beta pleated sheet, which literally look like sheets. And then you can't see R group interaction. Ribbon model, I would say, is fairly common when showing proteins. And then you also have a backbone model, which shows only the backbone and no R group interaction, kind of like your skeleton outline of the protein. And then also you can use more than one of these models when showing one protein. So you can use a ribbon model stacked with a spaceful model. If you need to see like a specific R group, you can have that R group in a spaceful model, and then have your secondary structure in ribbon model. So you can overlay these and use it however see fit for what you need. There's not, it's like, you can only use one for one protein. That's really it on protein level.
Does anybody have a question?
Okay. Now, protein structure. I'm gonna start obviously with primary structure, which is literally the sequence of amino acids and what that sequence is. From N terminus to C terminus, you're gonna have these peptide bonds holding together these amino acids. It's just the sequence of the amino acids, and like I said, the peptide bond is gonna hold those amino acids together. These bonds are super strong. They require a lot of energy to break. It's fairly uncommon for you to see like peptide bonds just spontaneously break in the cell. Like that usually shouldn't happen. Oh, if you change, like, let's say a question says, an amino, the 100th amino acid in a protein was changed from valine to glutamic acid. That's changing primary structure of the protein. You're literally changing the sequence of the amino acids, so you're changing primary structure. Whenever you have an amino acid change, you're gonna have primary structure change. Does that make sense? Any questions on primary structure?
And like I said, like previously, like peptide bonds are covalent bonds, and so they require a lot of energy and heat to break, and they're very strong. So for secondary structures. Yeah, okay.
Okay, your secondary structure is gonna be, like I mentioned when talking about the ribbon model, it's gonna be your alpha helices or your beta pleated sheaths. These are usually hydrogen bonds between the atoms. Austin is probably better to tell you the chemistry of it. But honestly, this is like niche basis. Hydrogen bonds are between the backbone of the amino acids. Your R groups are not going to participate in secondary structure. You're not going to see your R group bonding to the backbone. Doesn't happen. So like I mentioned earlier, your N terminus, your amino group and your carboxyl group are involved in backbone structure, and then that central carbon, that hydrogen attached to the central carbon. These are dipole interactions, and they're like that temporary particle charge. These hydrogen bonds are going to be weaker than your covalent peptide bonds. So let's say you increase heat in the cell, you know, obviously not like fatally to humans, but if you increase it enough, higher than your homeostasis temperature, you can break those hydrogen bonds and you can denature a protein. Denaturing of a protein is like rupturing the secondary structure in the hydrogen bonds between the backbone atoms. Okay, tertiary structure is going to be interactions between your R groups. So here you're not going to have your backbone interactions. This is just interaction of those R groups that I was mentioning, so your different amino acids. This is going to give a specific fold and shape and orientation to your protein and phase. This is what's giving it that fold and allowing like the protein structure is related to the function. So the way it folds the phase is directly related to how it functions in the cell. Yeah, between R groups, not backbone atoms. A lot of the bonds that Austin was talking about earlier are seen in tertiary structure, and they all depend on the properties of the R group. So I would just familiarize, like just get familiar with what kinds of bonds your R groups are going to make. Literally like sit down and categorize your amino acids. What kind of bonds are polar R groups going to make? What kind of bonds are polar charged amino acids going to make? What type of bonds? How are those bonds different? Why will like a polar charged amino acid make an ionic bond, but a polar shouldn't? What's different about them? Kind of get familiar with that. I feel like that's a common Laura question. She kind of likes the whys and the hows, and if I change this, what happens? So just get familiar with your bonds and what kind of bonds each like class of amino acid can make. So polar, non-polar, polar charged. And not always going to be true. Any questions about that?
Oh.
Hydrogen bonds. Also take this tertiary structure to, or
You can have, yeah, you can have dipole and dipole bonds in tertiary structure. Oh, structural ion dipole bonds.
I don't think she
Yeah,
does.
so just dipole dipole bonds in tertiary structure. Any other questions about tertiary? Yeah. Okay.
Yes, yeah, so in tertiary structure, I just wanna go over everything. The main type of, it's primarily charge interactions between the R groups that as they fold. And so just want to list the thing that we have talked about. So a hydrogen bond is a type of dipole dipole, which I talk about the water molecules over there. And then ionic bonds, sometimes it happens between like a charge amino acids. So this one and this one, it's full charge amino acids. So like, do you guys see this thing? There's like a plus charge here somewhere. Yeah, plus charge over here, plus charge over here, and negative charge over here, negative charge over here. That what dictates why they're in their place. And so as these two amino acid types would usually form an ionic bond together since they are full charges. So the next thing is van der Waals, like I've mentioned, the weakest type of bond usually happen between nonpolar R group. And the one very special type of bond, which is called a disulfide bond. It's a covalent bond. Happens so as we look in here, disulfide bonds happens between two cysteine amino acids because it has the sulfur here and the hydrogen here. There's like a reaction that catalyzes that bond, but basically it happens between two cysteine together. And so there could be one cysteine on one side of the amino acid and of the polypeptide and the other one at the end, and they could form together. And that's a disulfide bond, and it's covalent. Okay. The other thing is that disulfide bond doesn't occur in every single protein. It's like a special case. And so, yeah. Does anybody have a question?
And then I'll just add, like when I was talking about primary structure and I said, let's change one amino acid for another. So let's say a valine for a glutamic acid. Yeah, you're changing primary structure, you're also changing tertiary structure because this valine, it's not gonna make an ionic bond. It's the only real bond it's gonna make is usually like a van der Waal interaction, a London dispersion first interaction. But if you're changing it for a glutamic acid, you have this negatively charged molecule where you used to have a non-polar molecule. So this now can make interactions that it wouldn't make before. You're gonna change the actual shape of that protein in space. And if you're changing shape, you're changing function. Another water question, should we do that? Okay, and then quaternary structure, this is another thing that doesn't happen with all proteins, but quaternary structure is basically another chain of amino acids added to a chain of amino acids. So it's a bunch of polypeptides that come together and form a protein. One protein that has quaternary structure has multiple chains of amino acids. You can only see quaternary structure when you have more than one polypeptide chain. And the same bonds that you see in tertiary structure are involved in quaternary structure. Again, disulfide bonds are pretty rare, but they can happen. Happen in DNA damage. Your DNA is damaged. DNA forms a disulfide bridge. Yeah, this isn't like a part of every single protein, but it can happen. You only see quaternary structure when you have more than one chain of amino acids. And then here, like in this protein, you can see that this would be a polypeptide chain with an alpha subunit and a beta subunit. And this is another chain of amino acids with an alpha subunit and a beta subunit. So this is a dimer. Yeah.
Yeah.
dimer.
Just a fun fact, these heme groups carries oxygen. That's what, and hemoglobin proteins make up our red blood cells. And these like four parts there carry oxygen. And so that's why we need blood to live, basically. It's just a fun fact. It's not gonna be an exam. Is it? So one last thing, this environmental factors. Like I said, no, I didn't say about this. Salts are made out of like full charges, so NaCl, like mentioned over there, these full charges can interfere with quite a lot of non-covalent interaction, but specifically with like, yeah, they can happen a lot. So what that means is that
So, for example, what I mean is that, for example, if the water molecule like this, I love water. And this has like positive negative here, positive here. And then suddenly there's like a salt coming over here, which has like an Na plus here and Cl minus here. This charge interaction here will interact with this, and it can interfere with this bonding.
Another way it can do that is by interacting with those ionic bonds. So like your positive sodium is gonna come in and interact with your negatively charged amino acids. So it can disrupt ionic bonds too.
And the next thing is pH. pH basically means like the concentration of H plus ions in a solution, which you guys will learn more about chemistry, but it does have a significant effect on the chemistry of the body. The thing is like, if you guys have taken physics or even chemistry, there's like a wavelength line that like depicts like the energy level of each parts. Usually lights that have high energy wavelength can potentially denature proteins, basically add heat to proteins, which make the molecule vibrates, the molecules vibrates, and so the bonds are like shaking and then breaks it off. That's the same goes for heat. Heat adds in, it starts to get shaky, molecules start to moving around, and the molecules moving around makes the bonds start to move around and stuff. So about detergent, so as you guys get like what Jen said, like there's a
Phospholipid bilayer?
yeah, phospholipid bilayer. Basically, it has like a hydrophilic head and hydrophobic tail. That's why the tails wanted to be together because it's hydrophobic, it hates water. The head wanted to be outside because it loves water. And so a detergent monomer basically means a single detergent particle has the same characteristic and function like a phospholipid monomers, which basically also has a hydrophilic head and a hydrophilic tail. So what this means is that this chemistry of the detergent can interfere with the non-covalent interactions, similar to what ion does, similar to what ionic salt does. That's
I'll also add one thing about heat. Just because heat is bad doesn't mean that freezing cold is good. Your body has an optimal range at which you function. If you go too low, that's bad for you. And if you go too high, that's just as bad for you. It's kind of your homeostatic position. It's where you want to be. Your cells function best at like 37 degrees Celsius or whatever. So just be aware that like heat is bad because it'll break those bonds, and then freezing cold is also bad. So you want to stay at an optimal range, or I used to call it the Goldilocks rule. And, oh, there's someone.
This is like a mind map thingy I made. I spent 3 hours on it. So if you guys really wanted to like a summary of what I said, I can send this. So like send an email to me. I can send this, and if you guys want more like a detailed explanation, I have it too, and so please reach out to me.
Yeah.
Yeah, it will be there.
Yeah, Austin's your chem guy. I am not your chem girl. Hit me for bio anytime, but. And that's really it. Your problem set is due this Sunday, the 25th. So if you have any questions, I'll be here after, probably not until too late, but I'll be here. And then just email literally any TA ever. If you have any questions about the problem set, we're happy to help with whatever you need.
Also come to help sessions, because like the things that we cover here, we cannot go into very detailed stuff. That could